You know, there are things in our world that are everywhere, in the soil, in our water, and yeah, potentially inside of us. We're going to be talking about a microscopic parasite that has achieved just staggering global success, most of the time without its host ever even knowing it's there. So today, let's pull back the curtain on this master of stealth, Toxoplasma gondii.
And yeah, the answer is Toxoplasma gondii. This single-celled organism isn't just common, it's practically a textbook case of evolutionary success. In this explainer, we're going to break down its biology, its incredibly complex life, and its very intricate relationship with us, its human hosts.
And to do that, we've got a pretty clear roadmap. First, we'll get to know the parasite itself and its different forms. Then, we're going to unravel its wild two-host life cycle. After that, we'll dive into what happens during a human infection, see how we actually diagnose it, and then wrap up with how it's treated and, maybe more importantly, prevented. Okay, so let's start with the parasite itself. To really get a handle on its global impact, you first have to understand its fundamental biology and the different forms it takes on to survive and thrive.
Now, this might sound like a mouthful, but let's break it down. Obligate, intracellular, that just means it has to live inside the cells of another organism to survive. It can't make it on its own. And cosmopolitan distribution? Well, that's just a biologist's fancy way of saying it's found pretty much everywhere on Earth. A true globe-trotter.
So this parasite is a bit of a shapeshifter. It exists in three really key forms. First up, you've got the oocyst. This is the super tough, resilient form that gets shed into the environment where it can just hang out for a year. Then there's the tachyzoid. Think of this as the fast and furious stage, the one that's rapidly replicating during an acute infection. And finally, you have the tissue cyst, the dormant stage that literally hides out in our muscles and brain, just waiting. And here's the kicker. All three of these forms can infect humans. These different forms are all part of this brilliant, if not a little diabolical, two-host life cycle. And this strategy, needing two completely different types of hosts to finish its journey, well, that's the real secret to Toxoplasma's success.
And this slide really nails the core dynamic. The cat family, and only the cat family, is the definitive host. It's the one and only place where the parasite can sexually reproduce and create those hardy oocysts. Everybody else, rodents, livestock, and yep, us, we're just intermediate hosts. Inside of us, the parasite only replicates asexually and forms those dormant tissue cysts.
So how does it make that leap into humans? Well, there are a few main highways. The most common are either eating undercooked meat that contains tissue cysts or accidentally swallowing oocysts from, say, contaminated soil or unwashed veggies. Then there are the less common but critically important routes like congenital transmission from a mother to her fetus or through things like organ transplants or blood transfusions.
All right, so it's in. Now what happens? Well, the clinical picture of toxoplasmosis is really a story of two very different outcomes, and it all hinges on one critical factor, the strength of your immune system. For the huge majority of healthy adults, getting an infection is, well, it's rarely a big deal. Most people have no symptoms at all, or maybe it just feels like a mild flu that goes away on its own. The immune system is great at rounding up those tachyzoids and forcing them into dormant tissue cysts. But congenital toxoplasmosis, that's a whole other story. If a mother gets her first infection while she's pregnant, the parasite can cross the placenta, and the risk of severe damage to the fetus is highest during that first trimester.
And the consequences for these vulnerable groups can be absolutely devastating. Congenital infections can cause a classic triad of symptoms: severe eye inflammation, fluid on the brain, and calcifications in the brain. For immunocompromised patients, like someone with advanced AIDS, the real danger is reactivation. Those dormant tissue cysts can suddenly wake up and burst back to life, causing a life-threatening brain infection called toxoplasmic encephalitis.
So, given that this parasite is so good at hiding, how in the world do we find it? The whole diagnostic process is basically a form of biological detective work, using the body's own immune response as a trail of clues. This antibody response timeline is the key to the whole thing. Within a week or two of infection, the body starts making IgM antibodies. Think of these as the immune system's first responders. A little later, the IgG antibodies show up, and these guys represent long-term immune memory. So IgM suggests a recent active infection, while IgG, which can stick around for decades, tells you there was a past exposure. And that timeline tells us which test to use. Standard tests like ELISA are the workhorses for just finding these antibodies. But the IgG avidity test is incredibly important. It measures how strongly the IgG antibodies are binding to the parasite.
Low binding strength, or low avidity, means a recent infection. High avidity points to an older past infection. And making that distinction is absolutely critical when you're managing a pregnant patient, trying to figure out if she got infected before or during her pregnancy.
So that brings us to the final piece of this puzzle. Once we've found the parasite, how do we fight back? And maybe even more importantly, how do we stop it from getting in the first place?
Treatment is usually only for people with symptoms or those in vulnerable groups. The go-to combo is pyrimethamine and sulfadiazine. But because pyrimethamine can be harmful to a developing fetus, a different drug, spiramine, is used for mothers in their first trimester. And for babies with congenital toxoplasmosis, treatment is a major commitment. It often lasts for a full year.
And for those at the absolute highest risk of that dangerous reactivation, specifically AIDS patients whose immune systems are severely compromised, which we define as a CD4 count below 100, prevention is really the best medicine. Prophylactic drugs are given to stop those dormant cysts from waking up and causing that deadly encephalitis.
But when it comes right down to it, prevention is about a few simple but really powerful actions that break that chain of transmission. Cooking meat above 60 degrees Celsius, that kills the tissue cysts. Thoroughly washing your hands and your produce, that gets rid of the environmental oocysts. And for people at high risk, avoiding contact with cat litter is just paramount. These really practical steps are our best defense.
Which kind of leaves us with a final thought to chew on. Toxoplasma gondii is an ancient, unbelievably well-adapted organism. It has woven itself into our environment, our food web, and our very bodies. With no human vaccine anywhere on the horizon, its success raises this really profound question. Are we actually fighting an invader anymore, or are we just learning to manage a permanent, silent resident of our shared world?